Bioforest. Here we are in part 3 of neurons, and I wanted to show you this slide again to get a review of all these glial cells. So remember, lining a cavity are going to be these ependymal cells, and they're going to be able to secrete cerebrospinal fluid in spaces within the brain, and it's going to circulate throughout the brain, throughout the surface of the brain, and down into the spinal cord. Here are the astrocytes. So the astrocytes are going to wrap around a capillary and then wrap around a neuron, and in that way it's the king's taster. It's going to be able to filter anything that's coming from the blood to these cells. Here's an oligodendrocyte, and it's going to wrap around, forming the myelin sheath with spaces in between that are the nodes of Ranvier. And then also visible here are the microglial cells, which are the cleanup cells. And they're going to be mopping up any excess potassium or neurotransmitter that's still in that vicinity. So this is a really good picture there where you can see them all sort of doing their function, except for the Schwann cells that would be in the periphery. Moving on then, I'm going to skip this slide and remind you to always look at the external videos. There's a really nice one by Andres Lozano, and he's going to talk about deep brain stimulation. Very fascinating how we can actually treat with electrical activity instead of chemicals, and very, very interesting, so please watch that. Okay, the function of neurons, we've already said, is to conduct electrical impulses. The structure always has a cell body, a nucleus, a cytoplasm, and these cytoplasmatic processes, which are just the extension of that cell body, and those are called dendrites. And another type of cytoplasmatic process is an axon, and that is going to be where this electrical activity is transmitted down an axon towards another cell. So here in a diagram, this is kind of what you saw when you drew it. You don't really, when you get a cut on a microscope, you don't really see the whole thing, but here you could see the cell body. And the rest that you can see, sometimes you're uncertain whether you're seeing an axon or you're seeing a dendrite because you're unable to follow it the entire distance. But in this diagram, this would be the axon, and over here would be the dendrites. I wanted to point out the very different structural classification of neurons. Before we were talking about whether it was arriving at the central nervous system or exiting. Here we're just going to look at the morphology of them. Some of them don't have an axon at all. If it were to not have an axon at all, we're going to call it anaxonic neuron. A bipolar neuron, I want you to compare with a unipolar. They look very similar, but a bipolar neuron has dendrites at one end, this cell body smack in the middle, and then an axon on the other end. So dendrites, cell body, and axon. This one looks very similar. Dendrites, an axon, and a cell body, but notice how the cell body is off to the side. So it's sometimes difficult to see when this is just going like this, where it ends, where the dendrite ends and where the axon begins. And we would make that distinction sort of arbitrarily right by where the cell body is. This is a multipolar neuron, and this is the one that we think of most commonly with its dendrites and one axon. And then these are synaptic terminals, which means that's where it's going to connect to the next cell. And that next cell we're going to call postsynaptic cell. And then that would be called a multipolar neuron, okay, instead of a unipolar neuron. So where are these found? The anaxonic, no axon, is found in the brain, in the retina, and here are these extending dendrites. That's all you see. Bipolar neurons are also in the eyes. They're also in the ears. They're set up in very specific ways where they're going to connect certain cells that are receptor cells to certain portions of the eye, and I'll show you that when we get to the eye. For now, just know that these bipolar neurons are found in the eyes and the ears, and some in the nose also. The unipolar neuron is sensory And these are usually also found in some of the special senses. And we have these in the ear. The multipolar neurons are most common, and these are everywhere, these motor neurons, and they're everywhere, and they can have an extensive array of dendrites or fewer of them, or the arrangement morphologically can be quite different. This is a myelinated axon. We talked about myelin before, where you have that Schwann cell or oligodendrocyte that's covering it and leaving a space in between. We need these spaces here in between. I wanted to show you the difference between white and gray matter. So when we're looking at a wrapper like this, it's actually very fatty. So this myelin sheath is made up of fat, and because of that, it's going to look quite white if you look at it under the microscope. So this coating, I'm writing it out for you now, is white in color due to the fat. It's myelinated, and what happens is that in all myelinated neurons or axons, the conduction velocity increases with myelination, so it's going to be faster. Any axon that has that coating is going to transmit that electricity way more quickly. So most of our neurons are myelinated. There's very few that are not. Some pain receptors are unmyelinated. But in humans, most of them are. If you take a worm, no. But in humans, most of our neurons are myelinated. As an example, then, the unmyelinated ones would be pain fibers, where you feel that sort of pain that you can't really tell where it's coming from, and it's sort of diffuse. It's not this sharp pain. That tends to be an unmyelinated pain fiber. If I showed you a picture of this in the brain, you would see places that are full of white matter and others that are gray. So what's happening here? In this region, there's going to be lots of myelination. So this is going to be a region where there's lots of those axons that are traveling through the brain and are myelinated and are fatty. And in these regions that are gray, there's going to be cell bodies that are not myelinated and look quite different. Now, that was in the brain. If you look at the spinal cord and you did a cross-section of the cord, right, you just cut across it and looked at it, you would have then gray matter here in the center, and that's going to be a region again of cell bodies, and white matter, these are freeways of neurons that are traveling. Just imagine this long tube, right, which a spinal cord is, and things are traveling up and down that, and it's traveling here. In this region here, in this region here. Do you notice it's going to be there because that's where those myelinated axons are. Here too, all this. But gray then is going to be region of, I'm going to draw one, cell bodies, right? Draw another one here, cell body. There are some diseases of the myelin sheath, and a classic example is multiple sclerosis, which is an autoimmune disorder and very sad disease because it can start quite young at age 20 and up to 40 is the beginning of this. And it's where the individual forms, it sort of fights, your immune system fights your own myelin sheath, and it starts to degenerate. You sort of destroy it, and it's replaced by scar tissue. And what happens is that nerve conduction needs that sheath for that conduction to happen well. And once you start to destroy the sheath, then that conduction velocity drops, conduction at all drops sometimes. So there'll be muscles that are not innervated correctly and that don't respond to something that's coming from the brain because you're losing ions and that signal just isn't coming correctly to the muscle. This is an example of that. This is a sheath that would look intact, and this is a sheath that looks like it's been eaten away. So here we're going to have escaping potassium and escaping sodium, sodium, so that's going to be a problem. We're going to stop this here and continue on with talking about neurons. Thank you.